try to do this. So
Yep.
do you see something like?
We see your screen.
Oh, perfect. Yeah, thank you. Yeah, within next 15 minutes, I would like to drive you to other topic, which seems to be the core of this initiative, and that's towards biosensing, which somehow takes advantage of plasmonic nanomaterials. My name is Jakub Dostalek, and I work at AIT in Austria, and recently I've also some joint affiliation with the Institute of Physics, which indeed is not located in South Bohemia, but in Prague, but it's kind of over the border institution. What I would like to talk on is the work we did mostly in Thun, in Lower Austria, close to Vienna, where we have facilities at the joint campus with Boku University, and basically I'm operating within a group called Biosensor Technologies, where we kind of try to combine some biomimetic biointerface architectures with different sensing principles, mostly based on field effect transistors, some kind of electronic sensing and optical sensing, utilizing plasmonic materials, which I will focus more on this talk. Basically, the topics I'm interested about combine optical nanostructures, metallic nanostructures with specific plasmonic properties, with some polymer architectures, which then forms certain type of materials, which we mostly explore for the rapid analysis of species like biomarkers, pathogens, and we try to kind of utilize this type of materials to serve in tools for some life science research, related topics, and biomedical diagnostics. I'm going to talk on, not too many things, and I think most of them were done by these two people, by Stefan and Simone, and I'd like to acknowledge their work, not towards the end, but more in the beginning of the talk. And somehow to start with, I thought to use this slide I took from some other paper, which somehow illustrates the concentration of species one can find in bodily fluids like blood, and which can serve as markers, or which indeed can be detected to somehow diagnose, for instance, kind of infectious diseases. And one can see that indeed the concentration range spans over many orders of magnitude, and most often the biomarkers of diseases which are kind of established at something which people detect more on this kind of higher concentration range. However, as we now also see, for instance, in the context of the COVID diagnosis, the kind of detecting species which somehow relates to infectious diseases, one needs to get tools which are much more, let's say, sensitive, which somehow operates at this lower concentration range. And this is something which is done, but typically people need to use some techniques which involves enzymatic amplification of let's say assays, which let's utilize this this step, which makes the analysis not very practical if one would like to do this quickly on site. And obviously then we talk about techniques like, for instance, PCR, and like if I use this kind of COVID-19 example, then there are tools which are based on affinity interactions, like let's say immunoassays, which does not, let's say rely on this step, but then the sensitivity is not sufficient, and basically this, let's say the usability of these simpler tools is a bit under debate, let's say. And indeed, plasmonics and plasmonic structures offers means to somehow advance the sensitivity and efficiently probe species which can serve as biomarkers. In general, these kind of metallic structures, small metallic nanoparticles can support so-called surface plasmons, somehow optical resonances which are associated with collective oscillations of charge density eddies, which are accompanied with strongly confined optical field, which kind of emerges around. And the property of this field, I mean, how tightly we confine it, this can be tuned by design of the structures, and we can efficiently eventually probe species, which, I mean, let's say ranges in this size from few nanometers, like small proteins, to hundreds of nanometers, like, let's say, some lipid vesicles, like, I mean, exosomes, which can kind of shuttle cargo between cells or the whole cells. And this optical confinement indeed finds its applications in I mean, numerous optical techniques to probe biomolecules and let's say detect them. For instance, the technique of surface plasmon, I mean, resonance, and some types of enhanced optical spectroscopies like SERS, and let's say metal enhanced fluorescence. And somehow these structures, to let them serve in these techniques, need to be designed, and typically we kind of control the morphology of the structures to kind of spectrally tune the optical resonances, the plasmonic resonances in the spectral range that is needed, and also to tune, let's say, how tightly the field is confined. This is example of some arrays of cold particles where we see we can confine the field to distances of about I mean, 15 nanometers in the spectral range in the near infrared part of the spectrum. And this coupling of light to this, I mean, localized surface plasmons is spectrally, let's say, I mean, sensitive, and it occurs only at certain wavelength, and that's why these structures are typically can be seen as kind of, let's say, vivid colors when you attach them, for instance, to a glass substrate, like in this case. And yeah, in our lab, we'll kind of try to work with these structures. We use techniques which indeed Mikkel and Martin nicely introduced, including the nano imprint lithography. We did work in the past partially with EVG as well, and yeah, also with other colleagues from, in this case, Finland. I mean, in, let's say, VTT. In addition to, I mean, UV nil, we also do some optical lithographies to make the structures, which maybe we don't need to go too much to detail. And in some applications, people are interested in having kind of optical chips bearing these structures made in a way that which is, let's say, more cost efficient. And yeah, we explored approaches when these are introduced to some kind of flexible polymeric substrates and integrated with additional microfluidic devices to some kind of sensor chip cartridges, which then can be modified with bioactive fine molecules and directly used in the reader. And we also looked up with EVG a bit onto how to integrate these structures, for instance, at the bottom of microtiter wave plates. Another aspect I would like to raise attention to is how one treats the surface of these metallic structures. In affinity biosensors, one typically, let's say, modify the surface of these optically active structures with eye molecules, which can specifically capture target and light from the liquid you analyze at the surface, and then kind of efficiently optically probe it. And this can be done by, for instance, such polymer-based architectures, which is based on kind of thin hydrogel film with cross-linked polymer networks, which can be infiltrated with a large amount of these bioactive capture molecules. And indeed, this approach, the using of polymer networks, I mean hydrogels, which can form hydrogel, can offer a few advantages like this large binding capacity, anti-folding, biocompatible environment, which somehow repel absorption of other molecules we are not interested about, and can provide additional functionalities if we use, for instance, certain class of hydrogels which are responsive to external stimuli, which I would like to illustrate by a few examples. This polymer chains, which serve as kind of the binding matrix, can be covalently post-modified with the biofunctional molecules by using such amine coupling, or more recently we use click chemistry if we use some smaller molecules like short peptides. And in our lab, since many years, through which we let's say collaborated with the synthetic chemists, we have available kind of a toolkit of various polymers which are typically kind of tear or copolymers, where to the pre-synthesized chains we have already incorporated moieties we can use for the post-modification, and we like to make the polymer networks by photocross-linking. So we have these additional benzophenone or anthraquinone groups. And yeah, I mean, we can use some kind of synthetically made, let's say, polymers like this NABIAM or oxazolene, or some kind of more natural occurring polymers like this dextran. And I'd like to give, one example to illustrate how this optical structuring can serve for amplification of the optical readout of an assay. In this case, I would like to talk about the fluorescence enhancement when we design certain topography of the chip, which allows to resonantly couple light to surface plasmons at two distinct wavelengths. And the structure was designed so these wavelengths were coincident with the absorption and emission wavelength of the dye, which was used as a label in the assay. And by this coupling and both these wavelengths, we somehow amplified the fluorescence signal originating from the molecules which carry this tag with when kind of these molecules affinity bound to the surface. And in this slide, you can see the comparison of the fluorescent signal intensity, which was, let's say, measured in time upon titration of different concentrations of the inner light on the surface, which was flat, and on the surface, which was structured. And the structured surface was designed to provide this amplification of the, let's say, measured signal, and you can see increase by a factor of about 300, which indeed, if we then go for the small concentrations, allowed us to somehow push down the limit of detection to about sub 10 femtomolar concentrations, while on the non-structured surface, we've seen just flat and basically no signal. In addition, this is a bit linked to, I think, what will be the next talk about. We looked a bit on how the two-photon process could be used for the cross-linking of these polymer architectures with a precision which is at a, let's say, nanoscale. Regime. We thought to use, or we kind of explored how to use, for instance, the enhanced field intensity associated with the resonant coupling to, localize plasmons to kind of efficiently cross-link the polymer networks only within certain sub-areas around these, gold particles in the zones which are referred as to plasmonic hotspots. And eventually this could be used to then selectively attach the biomolecules to the most sensitive parts of the structure and eventually push the sensitivity even further. And in the last two slides, I'd like to address this possibility of using this responsive hydrogel architectures. And in this case, we thought to use the hydrogel matrix, which in the open, let's say, swollen state allows to capture the target in light, which can efficiently diffuse through the structure and then trigger the collapse, in this case by temperature, and compact the captured in light on the surface, and then eventually probe it more efficiently. And this kind of triggering of the collapse could be done for this anisopropyl acrylamide, so-called NABIAM based hydrogel by temperature changes around 30 degrees. And indeed, the system responds relatively fast at a millisecond time range, as we looked up in other study. And eventually, this slide illustrates that this allows to, in some way, a dual amplification strategy in the readout of the assay based on the fluorescence spectroscopy, when basically we use the optical, let's say, enhancement seen here as these peaks in the fluorescence signal associated with coupling of light of the excitation beam to the surface plasmons, and then eventually, if we trigger the collapse of the gel in which we capture the target in light, we see that the strength of these peaks, further increases by dragging the molecules closer to the surface when they can be more efficiently plasmonically amplified. And you can see that this, factor was of about 5, which we could put on the top of eventually this 300 I mentioned before. Again, I probably don't have so much time to go for details, and this drives me to the last slide. When basically in this talk, I tried to give you some a bit flavor of how plasmonic structures can be designed to serve for amplification of some optical spectroscopy signals like fluorescence, which can be used for readout of assays and serve for detecting of and the lights. And I showed you one example where we reached the enhancement factor of 300. And I also highlighted that indeed what's important is to somehow modify these inorganic metallic structures properly with biointerface architectures, which allows us to contact the chips to the biological fluids and capture the target and light from complex samples. And I illustrated that if you work this out together, you can reach rather low concentrations of analytes you can detect below 10 femtomolar, which are somehow, let's say, relevant to the demanding topics I introduced. Yeah, and these are the folks having fun after the work. And this is the very last slide. Thank you very much for your attention.
Thank you very much, Jakub. Yeah, we have already a question. Professor Klar.
Thank you very much for this presentation. A question, plasmonic biosensors are out in the market for decades. The gold standard is the BioCore system. Mm-hmm. So, how do your more sophisticated and also more expensive approaches compare to the BioCore system?
The BioCore system indeed offers tools which are optimized for biomolecular interaction studies, and it utilizes a principle of detecting of changes in the refractive index because of affinity binding of the molecules to the surface, which is kind of a attractive, direct, let's say, readout format, which doesn't require labeling of the molecules. In general, this technique, I think, is established in, let's say, research in this interaction. Studies. It's not much really exploited finally in the other fields which I introduced. For instance, it's not used for detecting really biomarkers in hospitals and so on. So I think, we talk about really different applications, and and I think it's a complex question which I'm not sure how to address in this very timely confined let's say frame we have here. But I think it's kind of different fields. Here we talk about which these techniques serve in. I hope I at least partially addressed the question.
Yeah, thank you.
Any further questions? I have a short question. Maybe I missed it. The hydrogel you're using, how is it applied to the surface? Is it?
I basically the gold surface is modified with this thiol having this photoactive benzophenone group, or it can be the other group, and then we typically deploy the polymer like these polymers in form of layers. So we kind of spin coat it indeed mostly, and then we can use some either we somehow irradiate the whole layer with UV light, or we kind of use some masks to crosslink only part of it, or recently we tried to explore some other techniques how to locally control, where we then finally crosslink the network and at the same time attach it to the gold surface because of this thiol SEM, which serves as a linker.
Okay, thank you very much. Good. Okay. I suggest we continue. Thank you very much, Jakob.
Thank you very much.
And we continue with Professor Klar. He is head of the Institute of Applied Physics at JKU in Linz. And yes, we can see your screen. Thank you very much. Okay, go ahead, please. Now we cannot hear you. Can we hear you? I cannot hear you, at least. Now we can hear you.
Yeah, okay, good. So thank you very much, and welcome to everyone. I think we are more than one talk behind, so I try to actually be short. I would like to talk a little bit about actually two-photon lithography, optical two-photon lithography. And to start with is a one-photon. So on the left-hand side, you see a Ablonsky diagram. So you actually optically pump in energy into, for instance, a fluorophore. Which is done here in the cuvette. There is a fluorophore in, and you excite it with 488 nanometers. And then you focus it, and in the focal plane, you have the smallest waist of the focus, but you see actually that all the way down the optical axis, you excite the molecules. And now Goeppert-Meyer introduced theoretically in 1931 two-photon absorption. And so when you use the same cuvette and you put in near-infrared light, then you see that only in the focal plane you excite the fluorophores, and that is used by two-photon microscopy to give you this nice effect that you actually really have a sharp view just from the focal plane. And so two-photon microscopy then was basically for bio applications invented 1990 by Denk, Strickler and Webb, and very short after, actually the same group, Strickler and Webb, just 1 year later, used it for two-photon polymerization, so not for imaging but for writing. And the same actually sectioning effect that you just watch in the focal plane in two-photon microscopy can be used in two-photon lithography that you only write in the focal plane. And then you can actually write three-dimensional structures, not layer by layer, which is usually done in three-dimensional polymerization when you do it layer by layer, but here you can do it in one go. So what is actually done is you prepare the monomers of your polymer, the monomers in a very thick layer, and then you can write in 3 dimensions in one go, not layer by layer. And so Strickler and Webb had a single paper in 91, and then later around 2000, Satoshi Kawata and his group stepped in, and they made, for instance, this very famous bowl here. It's a 3-dimensional, the so-called Satoshi Kawata bowl. So that is two-photon polymerization. And a few more examples here. And when we look to the Ablonsky diagram, so usually what is used is a negative tone radical polymer. In most cases, it's acrylates or methacrylates. And you need a starter, and the starter actually absorbs 2 photons, transfers to the triplet state, and from the triplet state you actually go to a radical, and that induces the polymerization of the acrylates or the thiolene or whatever you have. You can write nice castle structures, or here you can write a frog. That should be a frog. You actually see the voxels, okay? So you place voxel by voxel close to each other. The frog has actually a mistake, and it's good to have a mistake because you see that it is hollow inside. So it is really 3-dimensional writing, and you can have you can actually enclose something in a 3-dimensional manner. And a couple of examples from our institutes, most of them by the group of Johannes Heitz. So what they did is, for instance, they made a 2.5-dimensional grading in that case, and then they incubated this grating with fibroblasts, and you see that the fibroblasts actually tend to occupy these containers. So you can make a scaffold for cells, and later on in that project they also show that these fibroblasts actually can be convinced to carbonate, so to make a bone, a sort of a precursor of a bone. Another example is biomimetics, so bio-inspired polymer microstructures, and the biological model were bugs, and there's a certain species of bugs that has a microstructured surface on its back, and this microstructured surface actually directs the water towards the mouth of the bug. So this bug actually lives in a desert, and whenever there is water available, the bug makes sure to get all the water that actually condenses on the bug, because the water is directed to the mouth of that animal. And taking that as an example, we were writing these 2.5 dimensional structures, very similar to the biological original, and then down here, so this light gray area consists of that kind of structure, and depending on which direction these small parts actually point, when we apply an aqueous solution or some liquid, then you can direct the liquid to the right, or if you turn the whole thing down, you can turn the liquid to the left. So you can actually direct the flow of the liquid as it actually is done by this bug. Later on, or currently, we're now going to the 3rd dimension. So we have a 2-dimensional transport layer where we actually can apply a liquid, which in that case contains a fluorophore, Alexa 555. And on the right side here, we go into the 3rd dimension. So we place a micro needle, and this pyramid is also, the surface of the pyramid is also manufactured with a very similar surface structure. And the left pyramid, there's also a pyramid here, but it has a flat surface, so it doesn't have these microstructures. And then you apply this solution with the Alexa 555, and then you see actually that this solution creeps up this needle, which is microstructured, but it does not creep up the needle, which just has flat surfaces. And the project, which is FFG funded, that goes into the direction of micro needles. So the idea is to have an area with a lot of these needles, and instead of the Alexa 555, you apply some vaccine, and then you just put it onto your skin, and you get vaccinated. So you actually insert small droplets of the vaccine without having a painful syringe. Okay, so that is the microstructuring in 3 dimensions. And if you want to go even smaller, there was a problem, and the problem is the diffraction problem. So microscopy, the resolution in microscopy has been limited by diffraction. At least that was believed for a very long time. Ernst Abbe, who was a theoretical physicist in Jena, he actually got he was asked by industry, actually, by the Zeiss company, to think about the resolution in microscopy. Very famous, he found that the smallest distance that can be imaged in a microscope is given by the wavelength divided by 2 times the refractive index and the sine of the half opening angle. We all know that formula very well. And this is the diffraction. This is correct for diffraction, but the error was that he believed that diffraction limits the resolution, which is not true. It was Stefan Hell to point out that this is not true. And for fluorescence microscopy, as an example, so you excite the fluorophores, and in the lateral direction, that gives you, with the best objective lens you can get, it gives you a third of the visible wavelength, so let's say 200 nanometers. And when you confocalize it in the axial direction, it gives you the wavelength. So that's it. And if you have 2 fluorophores or 3 fluorophores in there, then all these fluorophores will fluoresce, and you cannot tell which one of those actually is fluorescence. That limits your resolution, unless, and that was the cool idea, you can switch off the fluorescence by some means, to the left, to the right, or above and below the focal plane. And if you can switch off the fluorescence very, very quickly, so you switch off the fluorophores before they can emit the fluorescing photon, then you are left over with a very small focus, and that should actually improve your resolution. And for that, he received the Nobel Prize in Chemistry in 2014. Does it work? So we need two colors. The first color excites your fluorophores. There's a measured point spread function in that case for the green color. And we have a fluorophore, and we have a 1 photon excitation, and we have a fluorescence, okay? And now the proposal was, if you add a second color, the red color, that actually induces stimulated emission. You actually use the excitation energy for stimulated emission, but you don't see any fluorescence anymore. And indeed, when we do the experiment, so we chop the red beam and we see the fluorescence being on and off, so you can switch the fluorescence. And a very important graph is this one here, where I show the red intensity and the remaining fluorescence. The remaining fluorescence is the tiny little bit of fluorescence here. And you see that as a function of the red power or intensity, you suppress the fluorescence in a very nonlinear way. That doesn't improve your resolution. So the original idea was to place this suppression in the outer rim of the focus, but not on top of the focus. And one can do that simply by playing around with the phase of the light, of the red light, which you put into the objective lens. And that allows you to create a focus that has a hole in the center. Okay, and now what is the size of the hole? The size of the hole, the half width of the hole, is of course given by the wavelength divided by 2. So that doesn't bring you much of an improvement, but there is this curve, okay? And at the maximum of the intensity, so you suppress all the fluorescence. At half of the intensity, you suppress all the fluorescence. At a quarter, at an eighth of the intensity, you suppress almost all the fluorescence. So if you actually fold this intensity pattern of the red point spread function with this fluorescence suppression ability, you blow it up, and you immediately see that actually the remaining center where you still have active fluorophores becomes very tiny, and one can quantify that. It shows that the traditional diffraction limit gets a prefactor, and the prefactor is essentially 1 over the square root of the applied intensity. And when you ramp up the intensity, you actually can improve the resolution quite a bit. It actually worked. So we have shown that around 2000, the confocal counterpart, and the same experiment when we switch on the stat beam. In those days, 20 years ago, we showed 100 nanometers in all directions. Meanwhile, the Stefan Hell group is down to below 10 nanometers in optical imaging. And now, so we have seen that we can use stimulated emission for depletion. That's it, stimulated emission depletion microscopy or nanoscopy to improve microscopy. Can we turn it to stat lithography? And the idea is we have a starter for the polymerization. So you actually absorb light, and then the excess energy of the electron is used for photochemistry, for instance, for polymerization. And if we bring down the electron fast enough, we can prevent photochemistry in the outer rim of the point spread function. And we have proposed that already more than 20 years ago. So the stat concept should be applicable also for 3-dimensional photochemistry. And it actually works. So I show you here some of our own examples. And this is a it's a door frame, and the door frame, freestanding in 3 dimensions, is written by 2-photon lithography. And then we switch on the stat beam, and we write these bars here. And from bottom to top, we increase the stat intensity. And you see that in 3 dimensions, using visible light, we get a bar that actually has a lateral and axial dimension of 53 nanometers. That is lambda over 10. We also can use, so usually we use acrylates, okay, acrylate monomers, and the gray here is our favorite triple acrylate molecule. And then we can actually use that acrylate molecule and mix in a few carboxy groups, okay, and we write every second bar here, and then we change the resist. And the new resist actually contains acrylate groups, and it contains thiol groups, and we write every other second bar. And then we can use Psi3 amine or Psi3 molyamide to have covalent bonds to the thiols or to the carboxys. And then we see that actually we can have successfully modified these bars with 3 different polymers in 3 dimensions. One example, one very recent example, again for a biomedical application, we had these flow cells, okay, we had a flow cell, and inside the flow cell we wrote gratings on posts. And the idea was the gratings on the posts, because when you have laminar flow has 0 velocity at the surface, okay? So if you want to do some kind of molecular fishing or something like that, we should actually have our structures that detect molecules inside a flow, okay? And the gray stuff here, here is assume the gray is a polymer that is pegalized, so it doesn't adhere to any polymers, or the other way around, any proteins don't adhere. But this very tiny little green spots, these are stat polymerized carboxylated acrylates, and they actually fish von Willebrand factors, okay? So we attach the von Willebrand factors, and then we can actually watch blood platelets adhering to the structure and forming very small thrombi. This is relevant for blood clotting. So I'm actually at the end already with my time. I acknowledge collaborations here at the Institute of Applied Physics by Professor Johannes Heitz and PhD students Christina Plamadiala, Richard Wollhofen, and Bianca Buchecker. At the Fachhochschule Oberösterreich, Jaroslav Jacek. Again at the Kepler University, Werner Baumgartner from the Biomedical Mechatronics Department. The blood clotting experiments were done in collaboration with Christian Gabriel from Boltzmann Institute. And the fibroblast experiments were done in collaboration with Innova Biotechnologies in Innsbruck by Rainer Marksteiner. Here is some of the latest reference we have, and funding was given by FFG and FWF and a few others, actually Horizon 2020. Okay, that brings me to the end, and I'm happy to answer questions.
Thank you very much. Yes, we have already questions. Please go ahead.
Thank you very much, first of all, for this wonderful talk. It was really, really interesting. I remember 2015, I read one of your first articles in this stat lithography. Can you maybe refer what is now the writing speed? What is the speed of this lithography technique?
Yeah, we are not in a mass production, so. If you want to mass produce, then stat lithography is probably not the right thing, because it's a step-by-step process. We are typically writing with 100 microns per second. This is a very typical writing speed. In most of the cases, I mean, you don't need 50 nanometer structure sizes everywhere. Okay, so in many cases, most of the structure you're very well done with a couple of microns of structure sizes, and only in very specific cases you would like to have the nanostructure. Like in the very last slide, so where I showed you that we have these very coarse gratings with these very tiny spots where we actually can attach down to single proteins. Okay, so that was the idea. We would like to have a very well controlled density of the von Willebrand factors. So yeah, it's good for prototyping. It is good for providing research samples to medical people or biology people. It's not good for mass production.
Thank you very much.
Good. Are there any further questions? Yes, Martin.
Hello. So I would have a quick question. It's understood that it's not fast enough for direct mass production. However, for the scales like we do with the replication of the nano imprinting and having an initial master, did you ever do such tests with your materials?
Not up to now. So to start, you need to start. We start with something. So to start with a imprinted rough structure, and then use that and put some tiny things on top of that, right?
Or yes, that could be basically two levels, that you have some larger structure, put then the small structures on top. But mainly I would be interested. Let's assume, so the slanting grating I've shown, it's not so easy also to make them now, especially in different directions. A single shot, if you could write something in a single shot directly, we could use it to replicate it immediately. We look into that with technologies that don't have the resolution right now, but that sounds very interesting because others are at the moment resolution limited to us.
Yeah.
To use this as mastering. So yeah, maybe we can discuss separately. But,
yeah, sure.
It might be interesting to give it a try if the materials withstand the replication process.
Yeah. Yeah, let's fine. Could be, yeah. Good idea.
Thank you.
Okay, thank you very much. I think we have to move on to our last talk today, which will, yes, here you are. You can see you, which will be given by Professor Stranianik from the University of South Bohemia. And we can see your screen. Perfect.
Now it's better?
Yes.
Okay.
Fine.
Good afternoon, everyone. And well, I will try to tell you something about our work and about our laboratory. The contribution I named as a nanostructured surfaces for bioapplications. And actually, this is related with our laboratory, which is the Laboratory of Applied Plasma Physics and the Nanostructures. And actually, here you can see in the central part an image of our building. And there are some stairs and the bridges. And this is something what can also explain what we try to do. So we try to bridge the material research with the bioscience. And actually here you can see also all the people that used to contribute in this type of the research. And if we are going to do more detail of what we are doing, so we are interested in some bioactive surfaces and materials. For example, we try to dope the thin films by the antibiotics, or we are dealing with the antibacterial surfaces. Further on, we are quite interested about the films for sensors and detectors, and we are dealing also with the nanoparticles and the nanostructures. Usually, we are applying those nanoparticles and nanostructures as a surfaces for MALDI, SALDI. I think that you already know. These are some kind of the advanced mass spectroscopy. Then was already mentioned some another sensing methods as SCIRs or LSPR. Actually, all this stuff of what we try to do here, and all these surfaces and materials are produced by the assistance of low temperature plasma, and then automatically we have to also include this in the investigation or in the frame of our interest. Hence, what we try to do in this part of the material science is somewhat illustrated here. So in our laboratory, we are dealing with several types of the discharges producing the low temperature plasma as an active medium. Then we try to deposit some films we use to analyze them, and then we try to optimize this whole system together with the input from the biological applications, and we try to tailor the films to produce the structures and the surfaces that are wanted. The possibility in our side are related with these techniques employing the low temperature plasma. And as you can see here, we are dealing with the homogeneous thin films. Usually these are in the thickness of the nanometers, maybe some hundreds of nanometers, usually not more than 1 micron. We can deposit the various materials starting from the metals up to plasma polymer. Further on, we are also dealing with the nanoparticles, usually, or the typical metal one, or so-called the core shell nanoparticles, combining the metal core and some, for example, polymer shell. Here you can see some histogram of the produced nanoparticles. Usually they are in the size in tenths of nanometers. Typical size is about 20, 30, maybe 50 nanometers. And then also by the graft and the substrate processes, we are also able to deposit some nano islands, some nanocomposites, and so on. We are just now, I will show you about some 3 fields where we try to be active and which we are the most interested in at this time. The first of all, we are dealing with the sensors based on the optical fiber. And there is a basic idea. It's pretty wanted that one sensor is able to combine 2 ways of the detection. And this is way what we try to do. We used to employ the optical fiber, which is able to combine the optical and electrochemical way of the detection. How it is done? The optical detection can be done, or for the optical detection, we can use the so-called lossy mode resonance. And it can appear, or it can be induced by the optical fiber if it is coated by some suitable film. In our case, it is the indium dope tin oxide. And actually, you can see it here. This is the fiber. The optical fiber is rid of the cladding. Here you can see the film that we used to deposit. Here is the bigger magnification. So finally, we have the film which is able to produce the lossy mode resonance way of the detection, the optical one. But this indium tin oxide, usually known as a ITO, is also the electrically conductive. So you can use it also as an active electrode for the electrochemical measurements. And practically the arrangement you can see here. So we have the optical fiber. By the light coming through it, we are able to measure the optical detection, and using the electrochemical compartment, we are able to diagnose or to detect some species also electrochemically. So one sensor in one time is giving us like 2 ways of the detection and 2 signals. That's a little bit more complicated. I don't want to go more to the detail, but for the lossy mode resonance, the optical way of the detection, we are working with so-called the modes of the resonance. Here you can see the basic mode and then the higher mode. Actually, you can see them here. And the principle of the detection is that once the optical fiber coated by the film is surrounded by some medium, then the refractive index is changed, and these resonance modes are shifting. And from this shift, we are able to or this is some sample in that way we are able to detect the species that are wanted. And for example, here you can see that the combination where we are able to combine the optical and electrochemical way of the detection. Here, the electrochemical detection is measured by usually by the cyclic voltammetry, and from the different currents then we are able to see what is the response on the current and on the species that are absorbed. The disadvantage of this method is the specificity, because with the surface is able to interact just only those species, or for example those biomolecules which are interacting with the surface. By other words, the sensors after the preparation by the plasma method is chemically functionalized. It's equipped, for example, with some grafts and antibodies that are able to interact with those species that we want to detect. But here it is somewhat illustrated and shown that we are able to combine those two methods, and we are able to confirm independently the species that we want. For example, here it was shown for the avidin. Further on, where we are interested or active now are some antibacterial nanostructured thin films. And these antibacterial, nowadays the antiviral surfaces are really highly wanted. That's due to the pandemic of COVID, of course. And there are some different strategies. The first strategy, how to prepare the antibacterial surfaces, are that they are antifouling. So it means this is usually quite closely connected with the high hydrophobicity that no species can be attached to the surface. We are doing something a little bit different, where we are using the chemical grafts or the films that are used as a scaffolds, which can serve as a buffer for some active species, for example, for the antibiotics. And here are already quite known approaches using, for example, tricalcium phosphate or some metal films that are porous, where the molecules can be impregnated. In our case, we were dealing with some different approach. By the plasma, we were able to deposit the plasma polymerized nylon by the sputtering of the nylon target. So practically, we have the nylon target igniting the plasma. We are able to sputter it, but we are sputter just only some fragments of this monomer chain. And these fragments are after that cross-linked in the plasma volume and are or on the surface, and then we are able to prepare the polymer which is cross-linked, where this coefficient we are able to tune by the parameters in the plasma. And further on, this structure we used to impregnate by the antibiotics. For example, in our case, it was the ampicillin. Then the ampicillin, once the ampicillin was stored inside, because of the natural amine grafts, we were able to coat it once again as some barrier layer, which allowed us to control the release of the antibiotics. This work is partially inspired by the coatings, for example, of some implants, where the antibiotics are stored exactly on the surface, or let's say in the surface, in the buffer of thin films, and then can be released with the defined time and with the defined amount. So that was done, and these are not just only the antibiotics that are providing some antibacterial effects. These are also, for example, some nano particles that we used. For example, the copper one or the silver one. These effects are already known for a very long time. But what we try to do is also shown here, that we used to deposit the nanoparticles, then we try to immerse them in some matrix Usually it is done again from some plasma polymer. For example, here it was from Teflon. By the way, we are also using the nylon that was impregnated by these antibiotics. And then we measured some response against some bacteria. There were done some different and various tests, but what is important is, or what is our challenge is to prepare the composites, for example, employing both the antibacterial metal species, for example, silver, copper, combined with the nylon matrix that could be impregnated by the antibiotics. What for it is, the motivation is that there are some different release rates, and then we try to prepare some composite or the matrix that is able to provide some antibacterial effect in some defined time. Further on, we are also dealing with these nanoparticles as a species for the detection. For example, as it is LSPR or SERS. Here you can see to show some screen from our former study where we study how the nanoparticles behave with the interaction of the laser beam. That was quite important for us when these surfaces were used for the MALDI. This is the matrix-assisted laser absorption, desorption, the spectroscopy. This is more or less, it is the mass spectroscopy, which is preferentially used for some larger biomolecules in bioscience, where these species are induced by the laser. So they has to be first desorbed, and then our nanoparticles serves as agents for the protonation, so giving the charge to the bigger molecules that can be detected in the mass spectrometry according the charge mass to ratio. And here, for example, we studied what is the effect of the laser beam irradiation, how the nano, and we found that the response of the laser is strongly influenced by the surface density of the nanoparticles. Once, if the nanoparticles are closer, they used to make the coagulation. They have the energy and they try to coagulate together, encouraged to make some friendship with the neighborhoods. While if there are some bigger nanoparticles with some lower surface density, they simply are not able to absorb all this energy, and they like explore. So then we are missing of them, and we observe the fragmentation towards some smaller species. But actually, today, I think that it was in the talk of Jakub Dostálek, we saw quite nice double peak SPR graph, which we try to do as well. For example, here, when we studied these two graded and two metal nanoparticle built surfaces. For example, here you can see what we try to do is the preparation of the composite, which consists of two nanoparticles, for example, silver one and the copper one, which we try to deposit in a really defined way. And for example, here you can see it that we were able to deposit some matrix or the nanocomposite with the defined composition between the silver and the copper nanoparticles, which was quite interesting for us, because in each point of the substrate we have the different chemical composition, and we were able to tune the chemical composition, but also the size of the nanoparticles. And then according to that, we were able to tune and manipulate with these absorption peaks. So we have like two independent peaks with which we're able to manipulate. And again, we try to employ it for the detection. So, and finally, we are also, that's the last slide, we are also interested in the industrial cooperation. Unfortunately, this part of the research is like partially, or that's not for the publishable results. But anyhow, we usually try to do something with the surfaces. These are some protective or tribological surfaces. Again, some functional surfaces, for example, for the grafting of the species on the surface, or some other projects that are employing the plasma, for example, as active agents for the cleaning applications. And in that way, I would like to thank you for your attention. Sorry, that shall not be here. Okay.
Thank you very much. Yes, we again have a first question.
Yes, I would have a question concerning the antimicrobial surface. The question for this ampicillin. Ampicillin has a moderate water solubility. I was wondering how long does it last, the antimicrobial effect, and how strong is it? Did you check this?
Yes, we did we did this experiment, and usually, that was here, I guess. Then we try to control the release by this protecting layer, or we used to call it as a barrier layer. So it means that according to the thickness, we are able to control the release. If you wanted to hear some numbers, actually, then if it is without the barrier film, then usually the release is within some hours. The bigger shock is, of course, at the beginning. Then we observed more or less the exponential decrease, and depending, of course, on the amount we found, that due to the thickness, of course, we are able to prepare or we are able to store more or less the ampicillin, which then can be released for some like days. Now, after 5 days, usually we with the thickness of the film like 300 nanometers, after 5 days, all the ampicillin is already released. And that was tested for some different bacteria, and that was quite effective. But once again, that's the motivation why we used to combine it with these nanoparticles, because there we observed the antibacterial effects for the longer time. So the motivation is like to have the first shock used by the antibiotics released from the nylon, and then followed by the release of the ions of silver or copper, or for example, like that. Yeah.
Thank you very much.
Yes, please, Professor Klau.
Yeah, thank you very much for your talk. So you have co-deposited silver particles and copper particles. My question is, can you deposit alloyed particles?
Yeah, this is actually what we try to do now. Yeah, we are able to do it because though these nanoparticles, once they are coming on the substrate, they need to lose their energy. So after the impinging of the nanoparticle on the substrate, then start to move around the substrate and interacting with each other. For the deposition of the alloy, we need to employ two sputtering sources, unfortunately. So two independent, one, for example, for the copper, then a second for the simpler silver, and then depends on the parameters how they coagulate together.
But I mean, without coagulation, so that you intrinsically produce alloyed nanoparticles?
Yeah, well, there are also, we haven't done it, but that was done, I think, in colleagues in Okinawa laboratory, that they are able to do it. They used to call it as a Janus nanoparticle, where they are, for example, like from two faces. Then it is done in that way that is a sputtered target already made from alloy or made like half and half metal, or even employing the two sputtering guns in one coagulation chamber. It is possible. We haven't done it here, but it is possible and already published. Yeah, so that's called as a Janus nanoparticle. Or what is more interesting for us are these core shell nanoparticles, where the metal core is covered, is coated, for example, like some sweets. So you have a nut and the chocolate.
Thank you.
Welcome.
Thank you very much. Are there any further questions? Yeah, so this doesn't seem to be the case. I will then say thank you to all the speakers, to all your contributions. I enjoyed it very much. Thank you very much, and I will hand over to Pavel for closing words.
Yeah, I would also like to thank to all the speakers for really excellent results and excellent presentations. I also would like to thank all the participants that you were joining this webinar, and we have some other follow-up webinars and scientific lectures. So follow us on social media, and I will also send some more invitations in the near future. So thank you and have a nice rest of the day.
Thank you very much.
Thank you.
Thank you.
Bye-bye.
Bye.
Take care.
Thank you. Bye.
Thank you. Bye-bye.
Thank you. Bye.